Hello, class. Welcome to part 2 of photosynthesis, chapter 13 in biochemistry. I already sent out part 1 of this lecture series of chapter 13 in the format of videos where we focus more on the plant anatomy, be it the thylakoid membrane, the thylakoid lumen, the stroma, and really we're just focusing on the chloroplast structure of where photosynthesis takes place. Today we're going to focus in on light reactions or light dependent reactions of photosynthesis as our second lecture period of chapter 13. You are the light of the world, said Jesus in Matthew 5:14a. That's our chapter theme, and I want to let you know that light is so important. Remember in Genesis, God said, let there be light. Light allows you to see things, but it also allows you to harvest this energy from plants. Okay, so plants harvest this energy, which then create small molecules, which animals eat, and then eventually we eat for our own energy source. And so light is so incredibly important, and without plants' ability to harvest this light for chemical energy, life would not exist on this planet. Here's an overview of photosynthesis. So photosynthesis, write this in, is one of the most important reactions on this planet since it causes plants to have energy and grow, which organisms eat for energy, which we end up eating for energy. And so essentially, hopefully you're able to watch some of the videos I put out, but one of them focused on what light is. And you may remember something called the electromagnetic spectra, okay, or spectrum. And what you have is different wavelengths, and they get shorter and shorter and shorter, and then they get higher and higher in energy. But in this one slice of electromagnetic energy, you see visible light. And visible light, appears like white light, but it's actually a combination of all the lights together reflected back at us. And we know that using prisms and other sorts of objects, you can divide them up into Roy G. Biv, red, orange, yellow, blue, green, indigo, violet. And that makes up your visible light spectrum. And so it's only a small fragment of the entire electromagnetic spectrum. But plants use this visible light for energy, or that energy, and they create chemical energy. So this visible light comes in the form of wavelengths and photons, which come essentially from the sun, okay, the fusion reaction from the sun. It makes its way all the way to the planet, and then there's certain photosystems in plants that's able to harvest this photon energy to convert it to useful chemical energy that the plants can have energy and grow, and that we as animals can also eat and grow and maintain metabolism. Okay, so hopefully that made sense. It's kind of, I guess we learned this back in elementary school, but I wanted to point you back to the matter of fact. Now that we've gone so much through our education, you can actually marvel at what's going on around us. Okay, so plants, which are called autotrophs, do not eat. Instead, they harvest their energy directly from the fusion-powered sun in the form of photons. All right, so you may recall that autotrophs, they're organisms that are able to form nutritional organic substances from simple inorganic substances such as carbon dioxide. And though we don't talk about that particular reaction today, that's reserved for the next video lecture, I did want to point out that autotrophs are able to synthesize their own energy from other sources, meaning like directly from the sun, from carbon dioxide, whereas a heterotroph, like ourselves, we're organisms that cannot manufacture our own food, and instead we obtain food and energy by taking in other substances, such as plants and animal matter. So I just wanted to draw that quick distinction for you. So here's my overview slide of photosynthesis. So I want you to know that photosynthesis can be divided into two different stages. And stage 1 is what we're going to lecture on today, and it's called the light-dependent reactions. Light-dependent reactions. And as the name implies, it depends on light to get through this stage. And the products of this stage are going to be used in stage 2, which I'm going to talk about in just a moment. But the reaction that you see in the light-dependent reactions is you take water, which eventually gets split during this stage, NADP+, you've seen that before, especially in the lipid chapter, chapter 12, ADP, adenosine diphosphate, plus an inorganic phosphate going to, and then what you get is O2, oxygen gas, You know that because we're breathing it right now and it comes from plants. You get NADPH, the reduced form of NADP+, and some ATP. Okay. Noting that the NADPH and ATP power the second stage, and the location of stage 1 is in the thylakoid membrane of the chloroplast. Stage 2 is called the light-independent reactions, and to me that is the best description of it. Light-independent. But sometimes you hear people refer to these as the dark reactions, or the Calvin cycle, which Calvin cycle is very appropriate also. But I'm gonna lecture on this in our next class period. But I do want to caution you of using dark reactions, because to me that's a little misleading. Yes, it is true that it does not require light, but no, it is not true that this only happens at nighttime. Okay, this happens during the day, the same time the light reactions are happening. It's just suggesting that there are reactions that are going on in plants, stage 2, that do not depend on light. So I think dark reactions is a little misleading, and I prefer light independent. Okay, so what you see here is you take your products, ATP and NADPH, which came from the stage 1 of photosynthesis, and then you combine it with CO2, and of course this is not a balanced equation, and what you yield is simple sugars, such as glucose. The location of this is the stroma of the chloroplast. So if you think about the chloroplast structure, the stroma is kind of that empty space outside of the thylakoids, and it has a lot of proteins, enzymes, it hosts a lot of other small structures. But for now, let's just consider it as a big goop that is kind of on the outside of your thylakoid. We'll talk more about this in the next lecture, referring to stage 2. So here's my way of depicting an overview of photosynthesis. So let's call this the light-dependent reactions, what we're talking about today. And what you input here is light, and that comes from the sun. And then you have water. This is exactly why you water your plants, right? It's why plants die without rain and all that. So just like we need oxygen to breathe, plants need water to survive. And what you get as your output of this is oxygen, O2. That's what we're breathing right now. Now, also the byproducts, as mentioned previously, were ATP and NADPH. What that goes on to is the light-independent reactions. Okay. Now, in the light-independent reactions, you do need ATP and NADPH, but you also need carbon dioxide. You've maybe heard it said we exhale carbon dioxide, which plants use, and they exhale, if you will, oxygen, which we use, and that's a nice little cycle going on. But this is carbon dioxide, CO2. And the output of this is sugar molecules. They're simple carbohydrates. For example, glucose. Another way to represent photosynthesis, this time showing you the cellular locations, is here you have the light reactions up here at the top left corner, and what we're looking at is a structure of a chloroplast, obviously highly simplified, but you input water into it, and then at the thylakoid membrane you notice that it takes NADP plus and a phosphate and a ADP, and you output NADPH and ATP, which then goes to the light independent reactions right here. Now, in order for the light independent reactions to work, you need CO2. Notice if you follow the arrow, and you end up getting some simple sugars with the base formula CnH2ON, which is a carbohydrate, CnH2O to the same N. And also after your light reactions, light dependent reactions, you have O2 as a byproduct. And you may notice these protons right here. Protons are necessary for these processes to work. So now I'm going to focus in on light-dependent reactions. So here's a nice overview of light-dependent reactions that I found on the web, so I gave Khan Academy credit for this. But I like that it shows you exactly what's going on and what I'm going to be talking about today. So you may notice at first glance that this looks a lot like the electron transport chain in the mitochondria, and you're absolutely correct. So because of that, I hope that this is pretty straightforward today as you engage with this material. So where's our location? We are looking at a chloroplast, and in particular, the thylakoid membrane. Now, you know what a membrane is, bilayer, organelle, all that stuff. We're looking at the membrane, and what you see is a lot of systems embedded within the membrane. Okay. Now, you notice one side is called the stroma. The stroma, I want you to maybe consider it to me, as I study this stuff, it reminds me a lot of the mitochondrial matrix in the oxidative phosphorylation sequence. And then you have the thylakoid lumen, the interior. This has a lot of semblance to the intermembrane space of the mitochondria. It's going to accept a proton gradient. It's going to be shot through ATP synthase to make ATP here in the stroma. Again, if you're struggling with the stroma is, you may want to go back and look at some of the videos that I've posted already. So here's what happens. You have this photosystem 2, which I'm about to explain what that is, and light enters it, which causes this photon to bounce across the leaf until eventually it hits this P680, meaning a photosystem that absorbs light at 680 nanometers. And once that happens, those chlorophyll molecules that make up that photocenter, or that core of this photosystem, release an electron. Okay, so the photon results in the release of an electron. That electron then gets transported to a mobile electron carrier, similar to ubiquinone in the electron transport chain in the mitochondria, which then goes to this cytochrome B6F complex. We'll talk more about that. But what I do want to let you know is because photosystem 2 just lost an electron, it needs that electron replenished. So in order to accomplish that, you notice right here in the lumen, or the interior of your thylakoid, you see this water splitting complex happen. And whenever you split water, you get 1/2 oxygen and 2 protons, so you're increasing that gradient. That electron then goes through that photosystem 2 and replenishes that lost electron derived from that photon there at that at that P680 photocenter. Now, as you may look at this reaction that's occurring at photosystem 2, the water splitting complex in particular, you may notice that it takes 2 water molecules to yield 1 breathable molecule of oxygen. So I did want to make you aware that you need 2 waters to get 1 O2. Once it goes to this mobile electron carrier, which is actually called plastoquinone, goes to that cytochrome B6F complex. It then gets the electrons go further to another mobile electron carrier, similar to cytochrome C in the electron transport chain of the mitochondria, which winds up eventually donating its electrons to photosystem 1. That electron gets bounced around, and you may notice that light can also hit photosystem 1 as well. What happens when light hits photosystem 1 is the exact same thing as whenever it hits photosystem 2. It bounces around, that photon bounces around until it hits that center, and that chlorophyll center loses an electron. Well, how does that electron get replenished? In photosystem 2, it got replenished by splitting water. Well, in photosystem 1, it gets replenished by this electron transport chain right here. Notice that plastoquinone, and then this one's called plastocyanin. It's another mobile electron carrier. So that's how this chlorophyll center gets replenished of its electron. Once it loses that electron after getting hit with the photon of light, it then goes to a series of complexes right here, and then it ends up reducing NADP plus to NADPH. Okay, and so you may notice that if you can follow my circles down here at the bottom left, oxygen is a byproduct and NADPH is a byproduct. All the while, if you were catching what was going on, we're building a proton gradient here in the thylakoid lumen, which then, just like ATP synthesis work, it has this proton conducting channel which sends protons through, and then it has this rotary motion that stitches a PI to an ADP, adenosine diphosphate, to make are energy-rich adenosine triphosphate, a molecule of energy, which is also needed in order to accomplish the reactions in stage 2 of photosynthesis. So, light-dependent reactions, an overview. I feel like I already did a huge overview, but let's look at a little bit more detail. So, as stated previously, this occurs in the thylakoid membrane. Looks like a little disc. Remember, a stack of those discs, they're called a granum, or multiple of them are called grana. It's a membrane-bound organelle that has numerous quantisomes, or granular structures that aid in photosynthesis, including photosystem 1 and photosystem 2. And so these quantisomes are just embedded within the membrane. You may want to look at a picture of what quantisomes look like, but the membrane's a highly folded system, and oftentimes you'll see things represented with like little triangles, circles, ovals, and all sorts of things embedded in the membrane. And they call these quantisomes, all of these together. And maybe the triangle represents photosystem 1, maybe this oval represents photosystem 2, so on and so forth. Maybe this little circle is just a light-absorbing pigment. And so that's what quantisomes are. In order to synthesize carbohydrates in stage 2, you do need the NADPH and the ATP derived from stage 1, which I'm going to be talking about in these light-dependent reactions. Now, a byproduct that's not used anymore in photosynthesis is that oxygen, okay? Oxygen, what we're breathing. It ends up leaving the plant cell via the stomata, which is just this tiny pore in the leaf structure that allows for gas exchange. I want you to think of NADPH as an electron carrier, similar to how NADH and FADH2 carry electrons in the mitochondrial electron transport chain. So remember, you can think of like NADP plus 2 electrons and an H minus. Okay, now you can split that one up into 2 electrons plus an H plus, and that's what I wanted to focus in on, is that a hydride essentially is 2 electrons. And then you can extract those 2 electrons to derive another proton, which can build a concentration gradient. To get started, I feel compelled to talk about chlorophylls first before we get into the photosystem. So let's talk about what a chlorophyll is. A chlorophyll, or chlorophylls, are green pigment molecules that resemble the heme. Y'all remember the heme, right? Such as in hemoglobin, it contains that central porphyrin ring. Now, heme in its porphyrin ring contained an iron, you may remember. Well, in this case, it contains a magnesium, Mg2+. Now, there's 2 chlorophylls we're going to talk about in particular, chlorophyll A. Chlorophyll A plays the primary role, okay? It's not a peripheral role. It plays a very direct role in photosynthesis. This is the one that actually loses the electron that gets transported through the electron transport chain in photosynthesis. So chlorophyll A is the primary key player in eukaryotic photosynthesis, as it's directly involved in the photochemical event, okay? Now, chlorophyll B also acts as a light harvesting pigment, passing along its energy to chlorophyll A. So though chlorophyll B is important, it's not directly involved. What it does is it helps chlorophyll A to be chlorophyll A. Does that make sense? And so there's a slight distinction there, but chlorophyll A is incredibly important. You're going to hear me talk more about it as this lecture progresses. I also want to mention carotenoids, kind of the orange pigments you see in many plants. It also functions as light harvesting pigments, so you can think of it like chlorophyll B in a way that it helps to guide light to these photosystems in order to accomplish photosynthesis. These proteins and pigments are all located in the thylakoid membrane. Okay, so you can find all these, which is why plants oftentimes have the green color, right? So here's the structure of chlorophyll. You do not have to memorize this, but what I do want you to see is the semblance to heme. So you may notice that here is a nice heme structure, the porphyrin ring. This is called pheophytin A. We're not going to talk much about pheophytin A, but it is involved in photosynthesis. Here's carotene, beta carotene. Notice it's highly conjugated, has a bunch of pi bonds, and as it turns out, those pi bonds do a good job at harvesting electrons and photons and reflecting and absorbing light. Okay, so it makes sense that you would want a highly conjugated molecule to do this. One word we might use for it is it has a chromophore. Okay. Now look at the chlorophyll B and chlorophyll A structure. It's hard to find what the real difference is. You notice that it's an isoprenoid. You can see that repeating isoprene unit, especially in this part of the molecule. Repeating isoprene unit. You notice that it has a little ester right here, an ester right there. Magnesium there in the center surrounded by a porphyrin ring. Well, I'm going to help you find the difference. It's just right here. Okay. Chlorophyll A has a methyl group where chlorophyll B has an aldehyde group in that same position. That's the only difference. It's oxidation levels. Okay, chlorophyll B is more oxidized than chlorophyll A. But recall that chlorophyll A plays a primary role, and chlorophyll B plays a supporting role. So I need to tell you what photosystems are before we get started. So a photosystem Photosystems are enormous complexes that are composed of proteins and light-absorbing molecules, pigments, you know that, it's the carotenoids and the chlorophylls and the pheophytin, that are keyed in to harvest light at designated wavelengths in the visible light spectrum. So remember, the visible light has typical wavelengths from about 380 to 800 nanometers. And that's where Roy G. Biv is located. We can't see outside of those wavelengths, okay? Everything appears invisible to us, like UV light, microwaves, and X-rays. But in these wavelengths, our human eyes can see these colors, Roy G. Biv. Now, these pigments are keyed in to take in light at those wavelengths, okay? And it utilizes it for chemical energy. And so that's what I wanted to talk about, these pigments, like I said, being the carotenoids and the chlorophylls. And these photosystems have these pigments embedded within them so that these photosystems can engage in these reactions. These photosystems have the capabilities of absorbing photons and transforming them to useful chemical energy. First up is photosystem 2. Now you may say, Morris, where's photosystem 1? And as it turns out, photosystem 2 was the second photosystem discovered, which is why it's called photosystem 2, but it's actually the first one used in the light-dependent reaction. So it's a little backwards looking, but they named them in terms of the chronology of when they were discovered rather than the chronology of how it actually undergoes photosynthesis. This is the larger complex. It's actually larger than photosystem 1. We're going to talk about photosystem 1 in a little while. Of note, oftentimes these are abbreviated as PS2 and PS1. The photosystem 2 contains pigments that harvest light at 680 nanometers. We call this photosystem 680. So you may see that nomenclature right there. And what it says is that whenever you get 680 nanometers of light, that's the particular wavelength that this photosystem harvests there at that photocenter. Okay, so at 680 nanometers, a reaction occurs at the core of photosystem, and the light harvested molecules there in the center are some specialized chlorophyll A molecules. Now I told you chlorophyll B and these other carotenoids, they play a supporting role of guiding the light there, but you can kind of think as a photon hits a leaf and then it eventually gets guided to the thylakoid membrane. You can think of the photon bouncing around, but it needs to hit this photocenter. And so eventually it hits it, and then boom, an electron flies off of it and then goes through the electron transport chain. Okay, but the photon needs to be guided to that photocenter, which contains the chlorophyll A molecules that specialize at a 680 nanometer wavelength light harvesting center. Okay, now the role of photosystem 2 is the oxidation, or it oxidizes water molecules, aka it goes from H2O to O2, or 1/2 O2, I guess, and donates the energized electrons to electron carriers that eventually reduce photosystem 1. Okay, so number 1, splits water give you H2, well, really two H pluses, and 1/2 O2. And then, of course, what came off of there are electrons. I'll just draw it like this, which then go to reduce the core, the chlorophyll A that lost an electron. It's now been oxidized, so now these electrons go on to reduce it again, so you can repeat this process. Okay, so this is one important role. It splits water to replenish those electrons that were just lost from the chlorophyll A reaction center, the P680. But also, you notice a little proton gradient also occurred, and what it also does is it moves electrons eventually to photosystem 1. Okay. Photosystem 2, it's a very large complex. And what it does is it spans the entire membrane. I'm going to show you a picture of it in just a moment. It has multiple subunits. You may want to go back to our protein chapter to understand enzymes and proteins, what subunits are. Okay, but what that's pretty much insinuating is that it has quaternary structure, and it's very big. And what it does is it has two specialized chlorophyll A molecules in the reaction center. Okay, and those chlorophyll A molecules lose electrons, which then pass on through the electron transport chain. The P680 passes the electrons to various electron acceptors through the complex. We're not going to get into the details like I did in the mitochondrial electron transport chain with like the NADH dehydrogenase complex, but just know that it gets passed through the complex due to varying electron affinities. But eventually, just like in the mitochondrial electron transport chain, it eventually found its way to ubiquinone or coenzyme Q. This one eventually donates them to plastoquinone, which is a coenzyme, right? And it's very similar to ubiquinone in the mitochondrial electron transport chain. And then PQ, as we call it for short, which is plastoquinone, it's the mobile electron carrier. It accepts one electron at a time, but transfers two electrons to the cytochrome B6F complex. In addition to the electrons, the plastoquinone picks up two protons from the stroma. Remember, I told you the stroma has similarities to the mitochondrial matrix in the mitochondrial oxidative phosphorylation. And what you end up getting whenever the plastoquinone picks up the two protons and the two electrons, you get the reduced form, the PQH2. PQ is called a mobile electron carrier or transporter. The electrons lost by photosystem 2 is replenished by the splitting of water molecules. We've already been over that. And splitting water molecules also causes a buildup of protons, or H plus hydrogen ions, which creates a gradient. You know where this is going, right? Because you saw the ATP synthase in the overview slide. But I did want to let you know you have to split two water molecules to create one molecule of oxygen. Now, this kind of has a reverse effect of mitochondrial oxidative phosphorylation, in particular the electron transport chain of it, because you notice that oxygen was the final electron acceptor, and you make water. Okay, this one, you split water to eventually make O2. Here's an actual depiction of what photosystem 2 looks like. I don't need you to know anything from this slide. I just wanted to give you a visual of how big it is, how it spans the whole membrane, though a membrane's not drawn, but you can imagine that's the membrane. It has various subunits, various domains, has a manganese cluster, not to be confused with the magnesium that's in the porphyrin ring, and a magnesium stabilizing protein. And so you see there's a lot of components needed, a lot of alpha helices, and so yeah, they were able to solve this structure. Pretty neat. So here's the overview revisited. Again, we're in the chloroplast, the thylakoid membrane. We've already seen how light is harvested in photosystem 2 as it comes in from the stroma, which is kind of the outside cellular space of the chloroplast. And the P680 photocenter, the reaction center, eventually gets that photon. It emits an electron, which gets passed, and now we have revealed the identity of that's PQ, but it obtains two protons from the stroma, so you can think of two protons and then it got those 2 electrons to make PQH2, which is the reduced form. I just told you that this is the cytochrome B6F complex. Okay, so that's where we are in this process, and because it lost its electron, as I'm circling right now, that electron needs to be replenished or else this whole thing would stop. And so you watered your plants, that's good. It's going to be able to continue this photosynthesis, so that water gets split into oxygen, electrons, and 2 protons. Protons building the concentration gradient, the electron going on to replenish the missing electron that is a result of the photosystem losing an electron due to the photon of light being directed at its core. Now, I'd mentioned that the plastoquinone, or rather the reduced form of the plastoquinone, donated electrons to the cytochrome B6F complex. Now, what the cytochrome B6F complex does is it accepts electrons from the reduced PQ, or plastoquinone, and then releases 2 more protons to the lumen. Remember, I told you the thylakoid lumen has similarities to the intermembrane space of the mitochondria, as the intermembrane space of the mitochondria accepted all the protons, which had the high proton concentration gradient. Okay, so this is further intensifying the gradient. You may remember why this is high potential energy, because you have a bunch of plus charges bumping into each other, and not to mention you have this chemiosmosis. It kind of high concentration wants to go to low concentration, and there's no way for it to happen. Okay, and so that creates a lot of potential energy as well. You may want to think of cytochrome B6F complex in a very similar way to the cytochrome BC1 complex of the mitochondrial electron transport chain. The cytochrome B6F complex is a dimer, meaning there's 2 monomers bound together, and each monomer contains 8 subunits, so it's pretty big. Okay, 1 subunit's already big enough. There's 8 of them, and since there's 2 monomers that have 8 coming together, there's 16 subunits in total. After traveling through the complex, the electrons are transferred to another mobile electron carrier. This one's called plastocyanin. Another carrier, and it carries 2 electrons at a time. So remember, there's 2 mobile electron carriers in the mitochondrial electron transport chain, that being ubiquinone, coenzyme Q, or cytochrome C. And so this one is kind of like the cytochrome C, except for, I believe it carries 2 electrons at a time instead of 1 at a time in the case of cytochrome C. Plastocyanin transfers its electrons to the photosystem 1 complex. Okay. Now we're finally talking about photosystem 1. You would think by the number 1 I should have already talked about it, but as I told you, they went by the chronology of when it was discovered. Okay, so this was the first photosystem discovered, but it's the second in the light-dependent reactions. Second photosystem, rather, is how I should have said it. It's smaller than photosystem 2 and contains pigments that harvest light at 700 nanometers, so we might call this photosystem 700, which occurs at the reaction center at the core of the photosystem, which are specialized chlorophyll molecules again. Chlorophyll A, chlorophyll B, but of course chlorophyll A plays the primary role in all this. The role of photosystem 1 is to energize and transfer electrons that are donated to NADP+. Okay, and you may recall NADPH and NADP+. This is nicotinamide adenine dinucleotide phosphate. So NADP+ is the more, it's the easier one to say. Okay. But it is a cofactor used in anabolic reactions, and you may see it in like vitamin B1 and all that kind of stuff. And anyways, it's a very important molecule. You saw it as a result of the pentose phosphate pathway previously in this class, and you saw it in the lipid chapter, like I said. And so, yeah, NADP+. So photosystem 1 is a very large complex, as you may imagine, and it spans the entire membrane as well, just like photosystem 2 did. It also has multiple subunits and 2 specialized chlorophyll A molecules at the reaction center. It has other chlorophyll A and chlorophyll B and carotenoids acting as what we call antenna pigments. directing energy towards the reaction center. So they kind of like what an antenna does in your car. It helps to receive things and direct it towards the source. And that's exactly what these act as, antenna pigments. They direct photons towards the reaction center, okay, the P700 center. Photons energize the reaction center and then transfer them to ferredoxin. Here's a new one we haven't seen yet. Ferredoxin. Ferredoxin then transfers these electrons to ferredoxin NADP reductase, easier to say as FNR, which after 2 electrons are transferred, it ends up converting the NADP plus to NADPH when combined with a proton. So here's what's happening, y'all. You have photosystem 1. It's kind of doing the same thing photosystem 2 did. It has these, it gets struck with a photon of light. It eventually makes it to this P700 rather than the P680 in photosystem 2. This is the P700 reaction center. And then the reaction center, the chlorophyll A, loses an electron. That electron then gets passed on to ferredoxin. Ferredoxin transfers these electrons to this NADP reductase. That makes sense, right? Because you're trying to reduce NADP to NADPH. And after 2 electrons are also transferred, you can make NADPH. And those protons you're going to see, they came from the chloroplast structure. Okay. And so here's a photo of photosystem 1. You don't need to know any of this, but I just wanted to show you that it does span the entire membrane, full of secondary structure. You see the alpha helices, you see the lumen right here, you see the stroma right here. The lumen being the interior parts of the thylakoid and the stroma being the exterior parts. And there's ferredoxin right here, willing and ready to receive the electrons derived from this photosystem. Here's plasticyanin. Remember, it donated electrons to this photosystem. Your P700 center lost an electron, so plasticyanin's electrons then replenish that. Plasticyanin's electrons are ultimately derived from splitting of water. And ferredoxin then goes on to that reductase, which then reduces NADP plus to NADPH. And finally, the ATP synthase, the end of this whole process. So we've already seen oxygen as a byproduct. That's a good thing because we breathe the stuff. You've already seen NADPH used, which is going to be used in, or formed, which is used in stage 2 of photosynthesis, aka the Calvin cycle, or light-independent reactions. And lastly, I told you we needed ATP to get those reactions going as well to make the small carbohydrates. So ATP synthase in photosynthesis is very similar to the mitochondrial ATP synthase. It contains a CF0 and a CF1 component. The CF0 component is the membrane-spanning proton conducting channel. And so that would be this part right here if you look over at the drawing. So that part conducts protons. Here's your lumen. So this is where all your H pluses are. Remember, you can think of the lumen as kind of like the intermembrane space of mitochondria. So it conducts protons. Proton enters. The shaft starts to spin. The CF1 component is the headpiece. So this part of the molecule, that's the CF1, located in the stroma. Okay, remember, you might think of this as the mitochondrial matrix part of the photosynthesis when trying to compare, and possesses the ATP synthesizing capabilities. So pretty much ADP, adenosine diphosphate, plus an inorganic phosphate, PI, go into ATP. And notice that you have this spinning shaft. You still have alpha and beta units, alpha, beta, alpha, beta, alpha, beta. And you have this stator that is holding the whole piece together. So it has a lot of structural similarities. And what this does, it takes that proton gradient, uses it to the advantage, and to make ATP. And of course, protons go over here in the stroma. And you've already seen previously in this slide that protons are absorbed from the stroma in order to make things happen. So in summary, you have photosystem 2 right here. And over here, out on the outside, here's the stroma. Here's the lumen. And light hits it. You see this light right here. And then it gets past until it finally hits this PS680. It loses an electron. And then it goes through this electron transport where it goes to plastiquinone, which donates it to cytochrome B6F complex, which then passes on to plastiquinone, or plasticinin, excuse me, which then donates the electrons to photosystem 2, which it gets hit with light. You see right there, loses an electron, passes it on to ferredoxin, which goes to the NADP plus reductase, which then synthesizes NADPH. And then since the lumen has all these protons, derived 2 protons, I do want you to know this, 2 protons from photosystem 2, a proton from plastiquinone, a proton from cytochrome B6F, and so yeah, you certainly have caused a proton gradient here in the thylakoid interior. I don't know if I emphasized this or not, but whenever we are reducing the NADP plus to the NADPH, the proton that combines with the electrons to end up forming the NADPH comes from the stroma part. Okay, so it's not coming from the lumen. And the stroma protons are coming from the ATP synthase. You can see how it can kind of make its way over there. The ATP synthase gives you the ATP, initially giving you ATP to the stroma. So I hope that all made sense. Of note, I wasn't sure if I said this correctly, but the cytochrome B6F actually donates 2 protons to the lumen, as does the reduced form of the plastiquinone, because you may remember that it had 2 H's on it. Okay. So here's your homework for this chapter. I want you to watch videos posted on the weekly to-do list. That is going to take a lot of time in and of itself, so take good notes from that video, as I do want you to have a robust understanding of this. And then here's your end of chapter problems. So this isn't for just this lecture, this is for the entire chapter 13 homework problems. Here's how I'm going to finish this lecture. Wonder rather than doubt is the root of all knowledge. This is a quote by Abraham Joshua Heschel. Think about that for a second as you look at photosynthesis. Fall in love with God. Okay, He created all this stuff. It's amazing that these huge protein complexes can be made just to pass along electrons in such an efficient and good manner that life persists on this planet. So you all have a blessed day. Reach out to me if ever you have any questions.